Welcome, everyone. I'm Anna Diaz, and it's my pleasure to kick off today's session. We're excited to have Cory Padilla with us, who will be speaking about the transformative impact of 3D genomics in oncology research. Cory will guide us through key topics, including the drivers of cancer, how the 3D genome reveals oncogenic mechanisms such as enhancer hijacking, and some case studies. He will also discuss Dovetail Genomics' unique solutions for oncology research. A bit about Cory. He earned his Ph.D. in cellular molecular biology from the Georgia Institute of Technology and holds a BS from the University of California, Santa Cruz. Cory joined Dovetail Genomics in 2017 as part of the R&D team and now leads product management. In his role, he bridges the gap between the scientific community and the company, identifying new research areas, gathering customer feedback for product development, and educating the community on the latest advancements from Dovetail Genomics. Feel free to submit your questions on the Q&A box, and Cory will address them at the end. Now, without further ado, I'll hand it over to you, Cory.
Awesome. Thanks, Anna. All right, so as Anna mentioned, today we're going to be talking about how 3D genomics can really help inform oncology research. And just a brief overview of what the talk looks like, we're going to start with an introduction to cancer and oncology. Hope you all in the webinar are aware of these things, and will be a very brief primer for you all. But I think it really sets the stage for how, in particular, 3D genomics can really influence our understanding of oncology and cancer genomics. And then we're going to walk through a couple of case studies that our customers have performed, which had some really nice outcomes and some really unique findings. And then we'll end with how we here at Dovetail Genomics are positioning our technology to help you guys better understand your particular area of oncology research. So, why do we care about studying about cancer and learning everything that we can about the cancer genome? Well, safe to say that we all understand that cancer is a leading cause of mortality. So this is a WHO data set about the global causes of death, and we can see that cancer comes in at number 2. So it's a really prevalent disease. It's a driver of mortality globally, and it's an area that we really need to try to understand to help treat and solve for some of these abnormalities that occur in cancer genomes. So cancer itself is hallmarked really by unchecked cell growth and in particular maintenance of the tumor microenvironment. So what are things that lead to that tumor growth, that unchecked growth? Well, abnormal cell proliferation. As things start growing, they keep growing, it kind of goes unchecked, and then you get to a physical tumor. The other thing is apoptosis resistance. So when things tend to go wrong in a cell's genome, it will oftentimes trigger a sort of cell death cascade, which will stop these tumor cells from growing and dividing. But if those particular pathways get shut down, then the tumor cells can continue to grow and divide unchecked. And then sustained blood flow to the environment, right? So as long as there's nutrients and oxygen getting into that system, those cells are going to thrive. And so it's really important for the tumor to maintain that blood flow to its system. And over time, what ends up happening is you get to metastasis, where these cells break off and start moving around throughout the body and really cause some really severe problems and often lead to mortality. So these are sort of like the key things that are driving tumor growth and ultimately metastasis. And so what's driving those things? It's typically misregulation of genes. So it could be across a wide range of things, but normally what it comes down to is there's some sort of variant or epigenetic feature that is turning some genes off and turning genes that need to be on off, or vice versa, genes that should be off back on. And so when we think about, the first scenario of just cells growing very rapidly in an unchecked way. So you have activation of genes promoting cell growth like RAS, inactivation of tumor suppressing genes like P53 or CDK, and then overexpression of transcription factors like MYC and GATA. So, all of these things tend to lead to more tumor growth than suppressing that tumor. When it comes to resisting cell death, it's the suppression of genes like BAX, which are really in part responsible for kicking off that cell death cascade. Also activation of apoptotic inhibitors like BCL2, and then P53 is also involved in the cell death pathway there. So you'll start seeing some patterns, and I think some of these genes should be very well known to folks doing cancer research by now. The RAS, the P53s, the MYC, CDKs, those things are all really important and what we look at when we look for misregulation in cancer genomes. And then when we think about sustained blood flow into that tumor, you think about genes promoting vascular growth like BEGF, and activation of vascular suppression genes like TSP1, and then again, cell signaling networks like RAS are pretty key in making sure vessel growth is occurring. And then when it comes to activation and metastasis, you're looking at reduction of cell adherence like cardigan, and then disruption of cellular matrix proteins like MMP2. And then you get also promotion of sort of cell mobility. So thinking about genes like snail and things like that get really kicked on. But really like I said at the start of the slide, it boils down to either genes getting kicked on that shouldn't be on and genes getting turned off that shouldn't be off, right? And there's diverse mechanisms for how you get these genes on and off. So there's a genetic component to this. So if you think about, genetic variation, either SNPs and indels, the small one to a handful of nucleotide events that alter the coding of gene sequence. So that can play a role there. It can also disrupt transcription factor binding if you're thinking about what a genetic abnormality can do in the epigenetic space. Then you have structural variation, where you can have things like gene fusion, which promote sort of atypical protein structure. Or you have things like enhancer hijacking, where after a structural variant, a oncogene is placed right next in a new regulatory network that kicks it on when it shouldn't be on, right? So those are how genetic abnormalities can really drive atypical expression. Then when it comes to the epigenetic space, this is a lot broader in mechanisms that it can really influence. So if you think about methylation, you can have gene silencing, or you can again have physical disruption of transcription factor binding sites, right? So in highly methylated spots, you might not get as much transcription factor binding as well as gene silencing. Then you have the actual chromatin itself. So you have nucleosomes positioned, and if they're all closed in together, oftentimes genes are not accessible by POL2 to being turned on, right? So if you have an oncogene and it's all blocked off by nucleosomes, it's not going to be expressed. Well, if something comes in and kicks out a handful of those nucleosomes or repositions them, then all of a sudden you can engage transcription by allowing POL2 to bind to the promoter site. And then again, these chromatin structures can disrupt transcription factor binding sites, which ultimately throws the regulatory network off. And then you have 3D chromatin structure. So this is really about thinking about how transcription factors engage with promoters to either turn genes on or disrupt gene expression. So you can have enhancer hijacking, as I just mentioned, as a part of structural variation, or you can have it as a breakdown or a gain of the three-dimensional chromatin structure that's in place. It doesn't have to be involved with a structural variant. And then you have these creation and disruption of chromatin boundaries that oftentimes influence regulatory networks. So one of the things I want to point out here is I know RNA is used pretty extensively to kind of try to understand gene expression and atypical expression in cancer cases, but it's really used to detect it and maybe to describe if it's on or off. But it doesn't really point to the mechanism of misexpression. You often have to go to the genome to understand if there's a genetic variant that's driving that misexpression, or you have to go to the epigenome to understand, is there something happening at the sort of transcriptional machinery, right? That structural engineering of how a gene gets turned on and off to try to understand the root cause of that misexpression. So RNA is sort of like an indicator, but it doesn't really tell you what's wrong that leads to that expression. So we're going to spend the next couple minutes talking about how, in particular, the 3D genome is really informative in the oncology space and why it's important to understand within the context of cancer biology. So the 3D genome has multiple different scales, right? If you think about how DNA sits inside of a nucleus, right? It's not all partitioned out like you'd see on a sort of karyotyping approach where you get the nice little chromosome 1, chromosome 2, and you get both alleles painted right next to each other. It's all jumbled up in there. It's kind of like a bowl of spaghetti that's encapsulated inside the nucleus. And that sort of structure has hierarchical folding within it. So you can have these sort of domains that interact more with each other than they do others. And then you can have looping events that actually bring specific gene promoters into the transcription factors that really recruit Pol2 and the other transcriptional machinery to turn a gene on or off. So that's one of the things that we really want to focus on here is that there's a widespread of features within the 3D genome. And it all comes from the fact that the genomes live inside of the nucleus. They don't live in your DNA extraction. Right? Once you extract that DNA and get it ready for sequencing, you lose all of that three-dimensional in situ orientation. And a lot of that is really helpful in understanding, again, how genes get turned on and off. So if we think about the very broad scale, we have these things called A and B compartments, where very open and active chromatin we consider in A compartments. These tend to cluster more with other A compartments than they tend to with B compartments. So one of the things to think about is if this little cluster here is an A compartment and this is a B compartment, even though in linear sequence space, these two regions are closer to each other, this region over here interacts more with this other A compartment than the two B compartments that might be closest to each other in that linear reference genome. So just because it's sequential in the linear genome doesn't mean it's sequential in the three-dimensional structure of it. And we could zoom in onto any one of these sort of compartments and we can see domain structure, where we see regions of DNA that cluster, again, within themselves more so than they do with adjacent DNA. And oftentimes we see in these domains sort of linked function genes or genes that are critical in maintaining a particular pathway. So if you think about cascade events where things need to be synchronized across different pathways, having the genes that matter in that pathway be sort of regulated by the same network is pretty critical. And then, as I mentioned earlier, you could zoom in even farther and see gene-specific loops where you have a gene promoter and enhancer that are being brought together by CTCF and cohesin inside of the nucleus, which allows Pol2 to recruit, and then you can kick off transcription. So these features are, in fact, quite important over the gene regulation or the influence that they exert over gene regulation. So what I'm going to do is for each one of those sort of hierarchical structures, I'm going to look at both breast cancer cell line and just look at what percentage of active genes really sit within these features. So in A and B compartments, again, these are very wide. We're talking, 10 to 50 megabase wide structures that exist inside the genome. About 80% of all active genes live inside these A compartments. And so if you have something that sits right on the boundary between going from A to B, or if you have a whole chunk of your genome switch from A to B, that can tell you whether or not there's a likely drop of expression there based on compartment change. Then when we get closer into those domains, these topologically associated domains or TADs, as you might have seen them in the literature, we see that like over 50, 65% of actively transcribed genes live in these TADs or at the boundaries of these TADs. And then when we get into loops, it's over 40% of the actively transcribed genes live in these sort of enhancer promoter loops. And there's probably more than that. You just have to maybe sequence a little deeper to capture them. But we do find that this mechanism is pretty widespread across genome and influences gene expression. So the 3D genome is really critical to understanding gene expression mechanism, and I'll show you why in just a very simple schematic. So these data give you direct evidence of in situ DNA to DNA interactions. And maybe if you're doing ATAC or CHIP or some other sort of sequencing approach that you do today, what you typically get as an output is like a BAM file or a bed file or a bigwig where you plot and you see one position and you see a signal on that one position indicating enrichment of some type of signal that you're trying to determine. With the 3D data sets, you get two positions and a contact score. So what it looks like if you were to visualize these data in IGV, you would see these arcs, and these arcs allow you to identify which part of the genome is interacting with the other, along with a score associated with that contact string. And so through this type of data, you can do some really powerful things. You can annotate variants of unknown significance, right? So GWAS data is just chock full of these things. To their cognate promoters. So a lot of these VUSs occur in non-coding regions, but it's clear that they're very consistent and show up in a particular strain of cancer, for example. You can look at accessible chromatin and regulatory networks, right? So if you have ATAC data, you likely know or have putative transcription factor binding sites, but you don't know exactly which one of those binding sites is talking to what promoter. So through these linkages, you could go from transcription factor to promoter. And again, if you're doing CHIP, like say if you're looking at something like PAX8 or something like that and want to understand how that gene or how that protein and its binding to the genome can influence gene expression, you can go and find all your PAX8 binding positions and link them back to their cognate promoters there. So with those sort of applications for the 3D genome in mind, we have a couple vignettes that are case studies that come from customer-based work that have really driven understanding of a particular type of cancer that they weren't able to see before without bringing in the 3D genome. So the first one is this functional annotation of GWAS and SNP data. So as I mentioned earlier, GWAS data is very full of these variants of unknown significance. And not only that, but they're also full of variants that are misannotated by saying, oh, this SNP doesn't necessarily go into the coding region of a gene, but it's really close to it. So they kind of assign it to the nearest neighbor promoter. And that's not always the best case because oftentimes even a shift in, a couple of bases can put you in a whole new regulatory network. So with the three-dimensional linkages, we're able to annotate and really describe which SNPs really influence the cancer progression. And then there's two different flavors of enhancer hijacking. One is through structural variants, as I mentioned earlier, where you have a translocation that puts an oncogene into an entirely new regulatory network. The other one is where you have a disruption in a chromatin boundary. So you lose CTCF binding, which allows a transcription factor to engage with an oncogene with no sort of genetic change, but simply through the loss of a CTCF binding, you are able to now bring an oncogene into a new regulatory network. So first one, the VUS study. So talked a little bit about this already, but it's really important, I think, to hammer home how important variants of unknown significance are, right? So more than 50% of the SNPs that we see in work that's been done are variants of unknown significance. And it's really challenging to annotate these because they often occur in non-coding regions, which kind of makes sense if you think about it, because there's only 2% of the genome that is really coding. The rest of it is non-coding, and trying to understand what a SNP and a non-coding region does is really challenging. That is why there is such a high percentage of VUSs in our GWAS data. And so one of our customers that was working in breast cancer really wanted to tackle this variant of unknown significant problems within the cohort that they were working on. So the problem that they had was they had about 150 hotspots where you have clusters of SNPs that were associated with breast cancer, but really, again, lacked that understanding of what the risk score was of those variants because they had unknown functionality, right? If you know what these SNPs are doing, you can do a better job of assigning that risk score to that particular variant. And so what I'm showing here is just a schematic of all the chromosomes and the sort of hot spots and how they're situated throughout the genome. And so what they did was they went in to the 3D genome data and did a hybrid capture against all gene promoters and went back and tried to link those gene promoters to the hotspots. And what they were able to find that on chromosome 10, there was a hotspot associated with an enhancer region that linked specifically back to GATA3. And this is really important for them. So now that they understood that there was parts or a handful of these cancer cases that had these linkages to this particular hotspot. And so now that they know that GATA3 is really the thing that is being influenced by these GWAS hotspots. And they went in and looked at the RNA and saw, sure enough, that GATA3 was misexpressed. And now they have a very concrete evidence for when they see this hotspot on chromosome 10 that it's influencing the GATA3 oncogene in this case, right? So it's a really powerful way to go about SNP annotation, particularly when you don't know the function of these SNPs. The next one is a customer that was looking at enhancer hijacking through structural variation. And so this has been around in the literature for a bit, where you have two different chromosomes, in this case, chromosome 14, chromosome 5, that have very separate regulatory networks. And what happens if there's a translocation event, you now have this fused translocation chromosome 14/5 inside of the cell. And what happens is in this particular case, the TLX3 gene, which is an oncogene, is turned off in normal scenarios, but after that translocation, now gets brought in to a network that has active enhancers that turns those oncogenes on. So what our customer did was they went into a handful of prostate cancer cases and were really trying to understand what was driving the ARID1A expression in prostate cancer. And they knew that in these cancers, the ARID gene was misexpressed, but they didn't really know why. And so what they really wanted to do is identify the mechanism of the oncogene expression in a handful of prostate cancers. And what they did was they used the 3D genome to detect a 1p36 inversion, which then enabled them to place it in the correct positioning that it is in the cancer cell, and look at the three-dimensional interactions and show that the DSP3 enhancer, which was on in the normal cell type, was being hijacked by ARID1 through the translocation, leading to oncogenic expression. So that's where that term enhancer hijacking comes from, right? We know that we have an enhancer that is linked to a particular gene, and after some sort of event, a oncogene comes in and basically commandeers that enhancer to influence its expression. And what we do see, which is interesting, is that the DISP3 gene, which was on in the normal case, was suppressed because it no longer had a strong interaction with its nearest enhancer. So that was it was a really nice paper that really hammered home the sort of enhancer hijacking case in terms of translocation events. And then the last translocation, or sorry, the last enhancer hijacking story really comes through that boundary disruption where you lose that sort of, or you lose the structure of the regulatory network that exists in normal state. And so one of the things that we do know when it comes to understanding gene misregulation is genetic variation only accounts for about 30 to 50% of oncogenetic expression, right? And that the epigenome, like you think about CHIP, ATAC, things like that, they do a really good job of mapping these regulatory networks, but they don't really tell us exactly what gene they're influencing, right? So we have this gap of we know that SNP or that variation doesn't always drive cancer, but we know that the placement of epigenomic elements within the genome don't necessarily inform which gene they're influencing. So instead of using things like co-expression, we can go in and really try to understand how these regulatory networks are influencing oncogenic gene expression. And so in this schematic here, we have, a gene 1, an enhancer, a gene 2, and its associated enhancer. And these are blocked off by those domains. So this is like a CTCF, maybe a CTCF and cohesin. That's really kind of blocking these two regions from interacting. But if you get a disruption in this boundary, now this enhancer can come in and talk to this gene and influence its expression that way. And there's two really great studies. The first one is looking at just cancer. And this was a really amazing study that was done where they saw that in this particular just cancer, there was a class of non-responders, despite having the exact same expression profile, right? So they do know that these were succinate dehydrogenase deficient just cancers. And they were giving the therapy to all of them, and only a handful of them were responding, the others weren't. And despite having the exact same expression profile, they couldn't figure out why that therapy wasn't working. And so what they did was they went into the 3D genome and the methylation signal and saw that there was a hypermethylation event at the CTCF binding, which a binding site which which kicked off CTCF from being able to bind. So it actually physically prevented CTCF from binding to the DNA. And that allowed for a super enhancer to come in and engage the oncogene and activate it. And so while this was probably in the other cases a genetic variant event that was causing this misexpression, that therapy was not helping at all for the disruption of the three-dimensional chromatin structure and the non-responders. And so they eventually went in and made a new therapy to go in and actually suppress that oncogene and kind of return it to that steady state. And so I think that's also something that's really powerful to think about in these data sets is these three-dimensional interactions are a pathway for new treatments and new biomarkers. So just relying on RNA itself to say, yes, it's misexpressed, doesn't necessarily do a great job at stratifying patients, and particularly in these subtypes of different cancers. And it's a really great biomarker. So now that we know, what's being disrupted, we can develop a biomarker to go help better stratify those cohorts. Then we have another case study where our customer was looking at multiple myeloma, and the gap that they really had is that they just didn't have a really robust catalog of the known drivers of MYC misregulation. So MYC is often misregulated in multiple myeloma, and they already had knowledge that there was a genomic component in terms of translocations and gene fusions, and in copy number variation that will give you multiple copies of MYC, and therefore you have double or triple the expression of MYC based on the number of copy number variants you see. But then what they went in and saw was that in the epigenome, you could also have a gene, not a gene amplification, but an amplification of enhancers. So in normal scenarios, there'd only be one enhancer engaged with MYC, but you can also have an amplification event that brings over the transcription factor binding site. So if you have more transcription factor binding, you're going to have more gene expression. So that's another way. And then you have these long range looping events where you have enhancers engaged with the promoters, and disruption or gain, in this case, gaining of loops, actually influence misexpression. So this was a novel mechanism for the myeloma phenotype for MYC misexpression. So again, it kind of speaks to the biomarker and therapeutic pathways that may arise by understanding and having a better catalog of the genetic or of the oncogenic drivers that may not exist just in the genetic space alone. So just to, I think, reiterate the value that the 3D genome has compared to maybe technologies that you're using today. So again, if you're using whole genome, RNA-seq, ATAC, or CHIP, anything like that to understand gene misexpression, what you get is one position and one enrichment score. And these are phenomenal tools. I don't want to underscore the value of these tools. They're great. They're great for building maps, of where SNPs and indels occur, for building maps of where regions of transcription are occurring or where misexpression is occurring. They're great at telling us where bases are methylated or where chromatin is accessible and where protein is bound to the DNA. But they don't really tell us anything about the genes that they influence. They just tell us the position. Where the 3D genomic data set, these genomic interactions with the two positions and one score, allow us to find and describe those linkages to really drive at that mechanism of misexpression. So again, annotating variants of unknown significance, really understanding linkages between factors, in particular misexpression, and really being able to build a robust understanding of what the gene regulation network looks like when you compare it with like the accessible chromatin information. So really, it's about driving home the mechanism, not the map. That's the difference. So how do we enable you all in oncology to use our tools? So as we talked about quite a bit, the application spaces that these data really play in are in academic research, translational research, clinical, and biopharma. So if we think about the case studies that we went through, thinking about how to understand these regulatory networks and functional annotation. You could do this in like neurological disorders across almost all cancers, leukemia, prostate cancer, breast cancer, myeloma, all of these things. They can be driven by three-dimensional genomic structure. Autoimmune disease, this is an area that's also growing in terms of thinking about how the 3D genome influences these autoimmune diseases, and then rare diseases and adolescent cancers. So these tools are really powerful at getting a really robust catalog of drivers of misexpression. And then as we talked about target ID, biomarker development, drug rescue, and cohort stratification, understanding that mechanism might allow you to stratify your cohort better. Understanding that mechanism allows you to build a better targeted therapy for that particular disease. So we have a couple of different approaches for getting at the 3D genome. So we have very global discovery. So it's basically whole genome sequencing, but in the three-dimensional space. You get the whole 3D genome structure. You can get all the features from AB compartments to TADs to loops. And it's a very unbiased discovery. You're basically casting a very large net and seeing what comes back in, and then you can work through that. Or we have targeted approaches. You could be interested in a protein. So you can, in the molecular biology, pull down on a protein of interest and ask, what is that protein talking to in terms of the surrounding gene structure? Or you can ask it from a gene-centric. So you could do a hybrid capture of the data set against all the genes and say, well, what are the genes talking to? And if you have, either CHIP or ATAC data, or you can go into databases like ENCODE that have some really nice curated known cancer enhancers and things like that, or you can go through and do some annotation that way. But what this does is it significantly reduces sequencing costs. It really improves your testing power because you're concentrating your sequence data all over a very small region of the genome. And then you can really increase your sample size to really kind of get at how the cohort structure really breaks out. So One of the things that we really think about at Dovetail is what is the value that we bring to you as a researcher? The first one is we capture more of the genome. So if you're doing whole genome sequencing today, our data set, or the data that we generate, covers the genome at the same level of uniformity that you would get with whole genome sequencing. So you're not missing any bases and you're not spending money over sequencing on stuff. Where other approaches, maybe you've heard of Hi-C or something like that, they're extremely biased. You oversequence parts of the genome, and there's a lot of the genome that simply isn't covered at all. So it's really hard to make true biological or have true biological insight if you're very biased in the data. And with that lack of bias means that we capture biology more accurately than the other approaches. So if here's your true biological signal, if you're looking at, say, for example, a protein binding site and the interactions associated with it, we recover that exact signal and we see enrichment around where there is interaction with the surrounding genome. Whereas with other Hi-C approaches in particular, they're very biased towards restriction sites because that's how they process the chromatin before doing the rest of the molecular biology on it. So you can really only put data where there's restriction sites. So you're going to distort, or in some cases even entirely miss, the true biological signal because you're biased towards where you can only put your data. Cost. We're really focused on reducing the amount of money you have to throw at these problems. So whether or not you're doing whole genome approaches or targeting approaches, we are up to 12 times less expensive per interaction, per looping interaction than the other Hi-Cs. So all in, our chemistry ends up becoming cheaper, and the amount of sequencing that you have to do is significantly less, in some cases up to half of what you would need to do for the traditional approaches to assess the 3D genome. And we really focus on scalable analytics, right? So we know everybody's computation infrastructure is different. We have very simple, straightforward, easy to follow guides for your informatic team to test and onboard the workflow. We also have services where you can send us the data, we can push it through our pipeline to get it back to you. And then if you're really new to the data, we also offer consulting where you basically get to use us as postdocs, obviously for a cost, but we're here to help and support you in your analytical journey as well. So I want to take the last couple minutes here to talk about what we have on the horizon. And if anybody's going to ASHG, we are going to be there, and we really want you to come and chat with us. We'd love to hear from you. And at that conference, we're launching our new LinkPrep technology to really bridge the gap between large structural variant detection and nucleotide level identification. So from SNVs all the way through structural variation. So these data type are really powerful at large structural variant detection, and they're also in its core whole genome sequencing. So that allows us to really span the full spectrum of somatic variants that you would be looking at in cancer samples. So here I'm just showing you, a schematic of how different technologies play out in the ability to capture different genetic variations, whether they be SNVs or indels, or all the way up through the big stuff that you would typically use, maybe FISH or karyotyping to figure out. But LinkPrep is a tool that spans that full spectrum. So the punchline here is that you'll be able to detect SVs with high sensitivity, but capture more classes of genetic variants than other technologies. And so what we mean by that is to really make the most use out of your short reads. So if you're using whole genome sequencing today, right, the LinkPrep technology captures SNVs, indels, and CNVs at the same efficacy as whole genome sequencing. So if here's the truth set of, say, like a cell line like K562, we're able to do exactly what whole genome sequencing can do at 30x sequencing. But when it comes to large structural variants, we have really, really, really high sensitivity. So at 30x genomic sequencing, whole genome sequencing maybe sees a little bit more than 60% of the variants that are there, but it also comes with thousands of false positive calls from the MANTA tool. But whole genome LinkPrep captures all of the SVs. And even if we downsample all the way down to 0.1x, we still see all of the SVs. It's not until we get super low in coverage to at 0.1x of the genome do we really start losing our ability to detect. So this is ultra high sensitivity SV detection with whole genome sequencing capabilities for the small stuff. And so what we're launching at ASHG is really improved whole genome somatic variant discovery through the Dovetail LinkPrep technology. As I mentioned, high sensitivity SV detection with base pair resolution, detect your SNVs and indels and copy number variation with whole genome sequencing-like capabilities. And to really, the aim is to really help improve the detection of somatic variation using standard NGS tools without buying new equipment, without going through different training, or without having to learn an entirely new data type to be able to describe these somatic variants. All right. Well, if there's no questions incoming from the group, please feel free to reach out and send your questions by email. I'm happy to answer any that we get. I think we can go ahead and close this thing down.
Yep. Thank you everyone for joining. Have a great day.